Walker. He is his exit seminar for his PhD. He's a molecular plant sciences graduate student here at SBS. So just a little bit of background about Berkeley. He got his BS degree in microbiology at Brigham Young University, and then came to Pullman, I think in 2007, to work for Decagon Devices, which is the instrumentation unit here in Pullman. And then fortunate enough for me, he decided to join the lab in 2008. So he came here about, I don't know, about 6 months after I came here. So we kind of started things together, he, I, and Jenny, as far as setting up the lab and getting things going. So that being said, Berkeley has done quite a lot as far as lab setup and getting things up and running in the lab. And he, I guess, sort of a testament to his ability in doing that is he's kind of the go-to person, especially when it comes to doing gas exchange in our laboratories. So with that, he will be sorely missed, as well as for his science as well. He's going to present the research that he's done for his dissertation. So he currently has one paper that he's a co-author on, already published, one that's in press, and 2 others that will be submitted shortly. So he's been doing quite well in the lab, and he's going to today tell us about some of the work that he's been doing on photosynthesis and the interaction with photosynthesis, nitrogen metabolism, and energy balancing. So with that, I give you Berkeley.
Hello? Is this on? Oh, good. So we're recording this for some of my committee members that are on off-site, so we'll have that. Thank you all for coming. So photosynthesis is the engine that drives life on this planet. Clothes you wear, the food you eat, the air you breathe comes from this primary conversion of CO2 to sugars using light energy from the sun. Now, it's not just humans, obviously, that rely on this. All ecosystems, well, at least the vast majority of ecosystems on Earth, are based on something green. Like any engine, photosynthesis is sensitive to the fuel that it's receiving and the temperature that it's running at. So climate change is changing both of these things for photosynthesis. With CO2, we're giving a high-octane fuel to photosynthesis, but at the same time, we're increasing the temperature that it's running at. So the big question is how well photosynthesis, this keystone of life on this planet, respond to the greatest change in climate in at least the last 11,000 years. These are the questions that I'm looking at. This is important to understand because we need to understand the response of net primary productivity to climate change. And we do this by looking at the response of leaf-level physiology. So I looked at this question in 3 ways. First, I looked at a process, photorespiration. And this, we'll talk more about what that is, but it involves a reaction of the initial enzymes of photosynthesis with oxygen instead of CO2. Second objective I looked at was the temperature response of rubiscokinetics. And then finally, I looked at how photosynthesis supplies the energy in the right kind and the right amount as needed for primary metabolism in the cell. So this idea of energy balancing. How do we supply energy demand with energy supply from photosynthesis? So again, photosynthesis is the primary conversion of CO2 into sugar using ATP and NADPH, 2 energy forms provided from the light reactions of photosynthesis. Right. So the initial enzyme of this conversion is known as rubisco. Now rubisco has a major affinity for CO2, but it also has a minor affinity for oxygen. When it reacts with oxygen instead of CO2, it results in the generation of compounds that are not able to be used in carbon fixation and need to be recycled to preserve carbon within the plant. Now this recycling is good, but it's not perfect. So this photorespiration is accompanied with a release of CO2. And this release of CO2 is not insignificant. In many plants, CO2 is lost through photorespiration at about 20% of the rate of net CO2 gain. So this is a huge loss of CO2 to the plant. Additionally, it requires energy both in the forms of ATP and NADPH. So my first objective, the hypothesis for my first objective is that this process increases even more with temperature than people think. Now, let me show you why we can hypothesize this. So photorespiration is a relatively complex process involving 3 organelles within the cell, involving about a dozen enzymatic reactions. So first we have that phosphoglycolate that's produced following an oxygenation by rubisco. It's passed through the peroxisome and into the mitochondria. Along the way, it's been converted to glycine, so 1 glycine per reaction with oxygen. So here we're looking at the result of 2 reactions with oxygen, 2 glycine. And in the mitochondria, it's decarboxylated and converted into serine. So here's where the CO2 release from photorespiration comes from. And it's released as 1 CO2 for every 2 rubisco oxygenations, or a stoichiometry of 0.5 CO2 per rubisco oxygenation. So that serine then is passed again through the peroxisome, converted to phosphoglycerate, which can then be used in the Calvin cycle. So this is where we get the carbon being recycled. But again, we're losing CO2 at this process. Now, as temperature increases, there's biochemical evidence that the amount of hydrogen peroxide within the peroxisome increases as well. This has the potential to react with photorespiratory intermediates within the peroxisome and release additional CO2. So not only is the plant potentially at high temperature bleeding CO2 through the mitochondria, but there's additional CO2 being lost through the peroxisome. So that 20% reduction in the efficiency of CO2 fixation may become a greater percentage as the plant is losing more CO2 per oxygen in. But my first objective was exploring this. Does the CO2 release of photorespiration increase with temperature? All right. Now let's shift gears from photorespiration to rubiscokinetics. Now, I'm sure the first question you're all thinking right now is, why should I care about rubiscokinetics? All right, let me tell you why. Yeah, nod, okay. The reason why is I'm going to tell you and I'm going to show you. So first I'm going to tell you, rubiscokinetics determine rates of net CO2 assimilation. This statement is incredible to me. You can have photosynthesis, a process involving probably hundreds of enzymes, intermediates, and regulatory mechanisms can be boiled down to the activity of a single enzyme. That's pretty amazing. Let me show you why I feel we can make this claim. By going forward. All right. So it comes down to rubiscokinetics. So let's talk about what kinetics are in general. I remember learning about this in undergrad, and I didn't understand the power of these models of kinetics until I started studying photosynthesis. So here we have the reaction rate of an enzyme, any enzyme. Here we have an increasing concentration of substrate. Eventually, when we get to a high enough concentration of substrate, we have a maximum rate of reaction. Now this is a kinetic value termed Vmax, maximum rate of maximum velocity of the reaction. If we go to 1/2 that maximum rate and go to the corresponding concentration of substrate, we get another value, Km, which represents the affinity of the enzyme for a reaction with that substrate. Now this is a really incredible tool. You can tell me the Km and the Vmax for an enzyme, and I can tell you what the rate of that reaction is at any substrate concentration. Okay, pretty cool. But how does that relate to photosynthesis? Well, our net carbon balance is based off of the rates of reaction of rubisco with CO2 and O2. And we can make that relationship with this equation here. So we have our net CO2 gain by the plant is equal to the rate of carboxylation. So this is the rate that CO2 is grabbing CO2, or the rubisco is grabbing CO2. We subtract from that the amount of CO2 that's being lost due to photorespiration. So here we have this ratio of CO2 release per oxygenation. From the biochemistry of photorespiration, we see that that ratio is 0.5. So we then know how much CO2 we're losing through photorespiration. And then we subtract from that again how much CO2 is being lost from respiration. So here we can use our rates of carboxylation, rates of oxygenation to determine our rates of net CO2 fixation. So now let's get to these rubiscokinetics. So without going into too much detail, you can see how we can determine our carboxylation rate and oxygenation rate from the kinetics of Rubisco for a reaction with oxygen, the kms for CO2, kms for O2, and the substrate concentration, CO2, O2, CO2. So we can plug all these into these equations, and we can get from fundamental biochemistry what our rates of carboxylation are, what our rates of oxygenation are. When we combine this all together, we get a unified equation. So this is just taking those equations you saw, and in this slide, we add to it the equations in this slide. So we're just putting all those together, and we get this equation. Now what this equation lets me do is it allows me to conjure a rate of photosynthesis, okay? All you have to do is tell me a CO2 concentration, and I can tell you the photosynthetic rate. That's a powerful tool. This is a tool used by people doing global carbon cycle modeling or people trying to understand what an individual leaf is doing. Now you'll notice that term alpha, which represents CO2 release for oxygenation, I don't see it in here. All right? The reason why you don't see it either is because it's rolled into this new term. This is called the compensation point. So the compensation point is the point where CO2 release from photorespiration is offset by CO2 coming in through Rubisco. So those two processes are balancing each other out. All right? So we can measure it using gas exchange, and it's equal to our CO2 release per oxygenation, a package of Rubisco kinetics, as well as the oxygen concentration. Okay? Okay, great, Berkeley. You have this model. There's a lot of models in biology. How many of them really work? Well, this one works pretty well. I'd argue that there are very few models in biology that work as well at this model at modeling photosynthesis. Here's an example. So here, I measured CO2 assimilation of a leaf, net CO2 assimilation. Here, I increased CO2 concentration, and here I have my measured response. Now this first section of line that you see, which models our CO2 assimilation rate really well, is that model that I just showed you. So that CO2, the model based entirely off of Rubisco kinetics. So now when you get to higher CO2 concentrations, you need one more model. It's derived in a similar way as the other one. You just have to make some different assumptions about what's available to Rubisco to do photosynthesis. So this model works. We can use Rubisco kinetics to determine rates of net CO2 fixation. Now, kinetics change with temperature. So to model CO2 fixation under a variety of temperatures, we need to understand how these kinetics are responding to temperature. All right? This has been measured in tobacco. So here we have Vmax, the response to temperature. Here we have Kc, response to temperature, Ko. So what you would do is, let's say you want to model photosynthesis at 35 degrees. You would go up, you would get your values for each of these parameters, you plug them into that equation, and you would get a really good idea of what photosynthesis was doing. And if you measured everything correctly, you would actually be getting exactly what photosynthesis is doing, because this is what defines photosynthesis. But again, we only have these parameters in tobacco right now. And people use these tobacco parameters to model everything from soybean to olive trees. So the next thing we need to understand is we need to understand how much CO2 is actually getting to Rubisco. So just because you have a certain CO2 concentration outside the leaf does not mean that will be the CO2 concentration at Rubisco. CO2 faces a tortuous path to get from outside to Rubisco, inside one of these cells. All right. So CO2 has to diffuse in through the stomates. This is a cross-section of a leaf. Once it's in the spaces between the leaf, it then has to go into the cell itself. And as it goes into the cell, it has to go through cell walls, cell membranes. It has to go through the liquid, all the guts of the cell, until it can finally get to Rubisco. So this second term, the diffusion of CO2 from inside of the leaf into the cell, is termed mesophyll conductance. It's represented by Gm. And another way to look at it is this the conductance? This would be the inverse of the resistance of the CO2 to get inside of the leaf. So our total mesophyll conductance is a combination of two parts of that conductance. And each of those parts has a different temperature response. So one of those parts, getting just through the membranes, has a positive temperature response. So this is probably an enzyme-mediated process. As you heat it up, those enzymes work more quickly. They transport more CO2 to rubisco. However, the diffusion of CO2 to rubisco does not really increase with temperature. It actually decreases a little bit. And so it's a combination of these two conductances that comprise our total mesophyll conductance. So again, this mesophyll conductance has been measured in tobacco. And we can see a positive temperature response. So this suggests that at least for tobacco, the transport of CO2 to rubisco is mediated by membrane-type processes. The problem is again, we only have this for tobacco. And people use these tobacco parameters for everything. So the question of my third objective is, are the temperature responses of rubisco and mesophyll conductance similar between species? How good an idea is it to use tobacco to model everything? So the last part of my dissertation focuses on this idea of energy balancing. So here's the problem. We have energy being generated in the light reactions of photosynthesis in the form of ATP and NADPH. That energy is used by the Calvin cycle to make sugar. Okay? There's other sinks for this energy, though. We know photorespiration is consuming a huge amount of this energy, as is the assimilation of nitrate. The light reactions involve some of the most powerful reactions in all of biology, using very reactive intermediates. If those intermediates do not have spent molecules of energy to put that energy onto, so if we're not consuming our energy at the same rate that we're producing it, then you have oxidative damage within the thylakoid membranes, and you basically fry the leaf. All right? So the leaf is in a precarious position where it can't produce or consume too much ATP, and it also can't produce or consume too much NADPH. And it has to do this in the face of tremendous changes in energy demand. So on a daily basis, we have changes in the rates of in the CO2 concentrations, in the temperature. We have daily fluctuations in how much nitrate is being assimilated. So how does the plant balance this energy supply with energy demand without frying itself? Well, let's take a look at what our total net energy demand is. So if you take rates of net assimilation and you break it down into rates of carboxylation and what you're losing in oxygenation, and you add up the energy requirements based on our knowledge of the biochemistry of CO2 fixation and photorespiration. If you add into that what the energy demand of nitrate assimilation is, Jerry Edwards actually published this a number of years ago, where the kind of average demand of a leaf is probably around 1.6 ATP per NADPH. This is under ambient conditions, and again, this is going to be changing on a daily basis, up and down, up and down. So let's take a look at what energy is being produced to see what our demand for energy balancing is. So here we have a thylakoid membrane. Energy production during photosynthesis occurs by transferring electrons and protons through this membrane. Right? It starts as we have water that is then split by light energy into protons, into oxygen. That excited electron then passes through the membrane, pumping protons as it goes down in energy. It's excited a second time, and it's used to charge NADPH. So this is where our NADPH is produced during photosynthesis. Then all those protons, they travel back through that membrane, through this turbine, ATP synthase, and they produce ATP. Now the way these reactions work, they produce ATP and NADPH in a fixed ratio. There's no flexibility in that design. So we have an ATP-NADP production of 1.3, but our demand is around 1.6. So where does the extra ATP come from? Cyclic electron flux is proposed to fill that gap. So cyclic electron flux takes electrons from NADPH, feeds it back into this proton pumping machinery, and we pump more protons per electron that's coming from water. That lets more protons go through ATP synthase. We have more ATP production, and we can increase the ATP to NADPH being produced during these light reactions. So cyclic electron flux is proposed to balance energy supply with energy demand. The problem is, when people take a leaf, when they change the energy demand conditions by putting in different CO2 and O2 concentrations, they don't see changes in cyclic electron flux all of the time. And as I did a literature review, I found that they saw it in situations where they were measuring under high light, but not under low light. Additionally, nobody had really formally modeled what the demands for ATP and NADPH were under their measurement conditions. And the last thing is nobody changed the nitrate availability to the plant as they measured cyclic electron flux. So my third objective was to really push energy demand. Can I really push energy demand and see changes in cyclic electron flux? And when does cyclic electron flux balance energy supply and energy demand? Okay, that's the introduction. All right, now we can get into the good stuff. All right, objective 1. Does the CO2 release of photorespiration increase with temperature? So one tool I used to look at this were photorespiratory mutants. So these mutants lacked key photorespiratory genes, and they had a disrupted photorespiratory mechanism. All right, so what happens when we mess up photorespiration? Well, here we have our photorespiratory CO2 release per oxygenation. Here we have oxygen concentration. And you can see that in these plants with disrupted photorespiration, they have a higher CO2 release per oxygenation, or that alpha term would be higher in these photorespiratory mutants. Okay, so this gives us a tool to compare to wild type. This gives us a plant that shows us what a plant looks like when it has a higher alpha, when it has more CO2 release per oxygenation. So the second tool I used was measurements of this compensation point. So again, you can see this compensation point are dependent on our CO2 release per oxygenation, or our alpha. They're also dependent on a package of Rubisco kinetics and oxygen concentration. And there's two ways to measure this through gas exchange. So one way, using oxygen exchange, assumes a value for alpha. So basically it measures all this stuff, and you have to give it a value of alpha. All right? Second method, when we use this LISC method, it measures the compensation point independent of any assumptions of alpha. So our strategy became really simple. We measure gamma star using a method that assumes alpha and one that does not assume alpha. We measure it in wild type and mutant plants. We measure it under ambient and elevated temperatures, and we ask the question, when are these measurements the same and when are they different? Let's see what that looks like. So here we have the CO2 compensation point. Here we have oxygen concentration. I left this equation up so you can reference it. You can see that we have a linear response of the CO2 compensation point measured without any assumptions of alpha. And we see we have a nice linear line. So this is what we'd expect. Our kinetics aren't going to be changing with oxygen. We see an oxygen response, so the slope then is proportional to this alpha. All right? So now, let's put up the measurements where we assume that alpha equals 0.5. We see that there are no differences between the two measurements of the compensation point at 25 degrees in wild-type plants. So this suggests that that assumption that alpha equals 0.5 is pretty good. When we put up this photorespiratory mutant that shows an increase in the amount of CO2 released per oxygenation, or an increase in alpha, we see it has a higher slope. So this is consistent with what we had measured previously at 25 degrees. That these mutants have a higher alpha. All right, now let's see what happens when we measure at 35 degrees. So the first thing you'll notice is there's an increase in the slope. Right? That doesn't tell us anything about alpha because, again, all of these kinetics are changing with temperature. So we need something to compare this slope with. So when we now compare it to measurements that assume alpha equals 0.5, we can see at every oxygen concentration we have a decrease in the compensation point. So this is evidence that the CO2 that this alpha increases with temperature to something higher than 0.5. Now, as further evidence of that, when we put up measurements of that mutant that has an increase in CO2 release per oxygenation, it's exactly the same as our values of the wild-type plants when measured under higher temperature. So this is good evidence that the CO2 release per oxygenation increases with temperature in wild-type plants. From this, we conclude that photorespiration appears to lose efficiency with temperature, and then also that there's significant differences between these two ways of measuring gamma star, and or the compensation point. As you'll notice from the previous slides, this is a real key part of a lot of these models. It's key for a lot of reasons, and this is an interesting point as well. So now let's move from the temperature response of photorespiration to the temperature response of rubisco itself. We're asking the question, are the temperature responses of rubisco and mesophyll conductance similar between species? So how are we going to measure rubisco kinetics? So there's two main ways to measure rubisco kinetics. One is an in vitro method, Latin for in glass. One is an in vivo method, in living or in life. The in vitro method, we take a leaf, we juice it up, we put it in some kind of buffer where we assume we have the right pH, the right CO2 concentrations, the right cofactors associated with the enzyme, and we measure the kinetics that way. Or we can take a leaf, we can clamp it into a machine that measures the CO2 gas exchange of the leaf, and then use that relationship between rubisco kinetics and net gas exchange to then infer what our rubisco kinetics are. So if you were going to pick a way to represent rubisco in a living system, you'd probably want to use this in vivo method. And that's what we use. We also measured mesophyll conductance. We were looking to see if at least between the two plants we measured, if they had an increase in mesophyll conductance with temperature, or if it was a more flat response. That could give us an indication of what processes are dominating CO2 transfer. Now the plants. So one of the first things Emily asked me when I told her I studied tobacco is she asked me, why are you studying tobacco? All right, so I mean, I don't think about this because I know tobacco. And my work is not financed by Philip Morris. All right, the reason that tobacco has been studied is it has these nice big flat leaves. Those are really nice when you're trying to measure gas exchange. Big surface area, real flat. They're pretty simple genetically. So that's why work had been done in tobacco. What I compared it to was this plant Arabidopsis thaliana. So Arabidopsis is a simple plant genetically. It's a big model organism for a lot of plant physiology, and it's easy to grow. Two other reasons why I picked these plants. One of those other reasons is tobacco is a more warm adapted plant, right? Whereas Arabidopsis is more temperate adapted. So maybe there'll be differences based on adaptation, something that we can start approaching. The third reason is to measure the in vivo rubisco kinetics, you need to use plants with a genetically reduced amount of rubisco. So you have to use transgenics. So these two plants we had mutants available for, so I was able to use them to measure these in vivo kinetics. So let's take a look at what it looks like. So here we have the temperature response of 5 key rubisco kinetics. Compensation point, VC, VO max, KC, KO. So again, we can go to any temperature, plug it into this equation, and get rates of net assimilation. So first I'm going to show you what I measured for tobacco. We'll compare it to what other people have for tobacco. But we can see I'm pretty similar. My temperature response for Tabasco is pretty similar to what other people have measured. Great, I'm measuring it correctly. Now let's put in the temperature response of Arabidopsis. So just taking a step back, there doesn't seem to be a whole lot of difference. So maybe there's some difference here in the compensation point, but statistically, there's no significant difference between the temperature response of any of these individual kinetic parameters. But we're plugging these kinetics into one equation to get answers. So what if there's subtle differences between these kinetic parameters that result in maybe a larger than you'd expect change in net assimilation or predicted net assimilation? So to test if this was the case, I represented photosynthesis either using the kinetics of tobacco or of kinetics of tobacco or of Arabidopsis. So here's the modeled photosynthesis at 35 degrees. Here's CO2 assimilation rate. Here's CO2 concentration. Here's Arabidopsis in solid, tobacco in a dotted line. You can see at the extreme CO2 concentration, we don't see a big difference in CO2 assimilation. Right? But when we move to these intermediate CO2 concentrations, where photosynthesis occurs on this planet, at least, we see that there can be maybe a significant difference in there. In fact, there can be up to a 10% difference between modeled photosynthesis based on which rubisco you're using, rubisco from which species. So that can potentially impact your models of photosynthesis. Now let's take a look at mesophyll conductance. So here we have mesophyll conductance, here we have temperature. When we look at tobacco, we see that same temperature response that other people had measured. However, when we look at Arabidopsis, we see that there's no temperature response. It's pretty flat, maybe even decreases here towards the end. So this tells us right here that there are differences in the temperature response of mesophyll conductance between species. If we break mesophyll conductance down into membrane diffusion and into the liquid diffusion, we see that where tobacco seems to be dominated by the temperature response of membrane diffusion, Arabidopsis is better represented by liquid diffusion, simple liquid diffusion through the cell guts. Right? So it would be really interesting if this is something that's generalized to different kinds of plants adapted to different environments. So that's a direction of future work in this. But at the very least, what's exciting about this is tobacco has a way to get more CO2 to rubisco at elevated temperatures. So a lot of crop plants are more temperate adapted. So this may be a route to improve the transport of CO2 to rubisco if we can figure out how tobacco manages this increase. So our conclusions here are that rubisco kinetics vary in temperature response by species, temperature response of mesophyll conductance varies between species, and there are different mechanisms that dominate CO2 transfer based on what species you're looking at. All right, so now let's shift from temperature response of photorespiration or rubisco kinetics to this idea of energy balancing. And asking the question, when does cyclic electron flux balance energy supply and energy demand? So our approach here is pretty simple. We change energy demand, we put the leaf in different conditions of energy demand, then we measure cyclic electron flux. And we see, does cyclic electron flux correlate with changes in energy demand? So we can use those biochemical relationships of gas exchange to rubisco kinetics to determine what our rates of carboxylation and oxygenation are in a given CO2 and O2 environment. We can then add in nitrate assimilation, and we can use what we understand about the biochemistry of carbon fixation and photorespiration and nitrate assimilation to calculate what our total ATP and NADPH demand is under these different measurement conditions. We can then compare what our demand is to what's being produced in the light reactions and determine how much extra ATP we need and how high of a rate of cyclic electron flux we would need to support that rate of this given energy demand. Okay, so we can see I picked several different treatments that create a nice spread of demand for cyclic electron flux. So how do we measure cyclic electron flux? Well, when we have high rates of cyclic electron flux, we have more proton flux per electron flux from water. So when we have more cyclic electron flux, we have more protons per electrons. And we can measure this through spectral signatures of photosynthesis. I'll show you what that looks like in a minute. So our approach then is change energy demand, then we measure the ratio of proton flux to electron flux. So I grew all my plants hydroponically. I learned a lot about hydroponics, and there's a lot of great resources online about growing plants hydroponically. I grew them hydroponically because if you're trying to change nitrate supply to a plant, it's really hard to do that in a soil matrix, right? I mean, how do you know you got all the nitrate out? I mean, maybe you water it for a week and then assume that it only has nitrate available. No, with this, we can take the plants directly from a complete nutrient supply into one that just has nitrate or doesn't have nitrate. But we measured them, I measured them in this system here. So I never thought of a good name for this. Call it Frankenstein. The basic idea is we take our plant, so that green thing, that's our plant. We then put it into this temperature-controlled cuvette here. We can change what the CO2 supply is through a mixing system that was under here. Change rates of CO2 fixation. We can modulate rates of photorespiration by changing the oxygen supply. And then we can change what our nitrogen supply is. So we can either give it nitrate or not give it nitrate. When I didn't give it nitrate, I gave it ammonia. That doesn't require energy at the leaf level. Then the leaf is clamped into this spectrophotometer. Let's take a look at what this looks like. So Lauren will recognize this picture. Here we have the CO2 and oxygen being provided to the leaf. It's in between these two light guides, and the spectrophotometer blasts that leaf with light of different intensities and different wavelengths, and we can pick up what those spectral signatures of photosynthesis are. So I can measure then the rate of proton flux to electron flux. So let's take a look at what we saw. So I measured under low light and under high light. We'll look at low light first. So here we have our rate of proton flux. Here on the x-axis we have our rate of electron flux. The slope then is the ratio of the two. So that slope should represent cyclic electron flux. So here are two lines. These lines model the extreme differences in energy demand, or demand for cyclic electron flux, that I put my leads under. So here would be the low, and this dotted line would be the high. Now this is what I measured. So this is under all those conditions you saw on the table. One thing you notice is that there's a linear relationship between all these points. That's good. That indicates that there's a constant rate of cyclic electron flux under all these conditions. The other thing you notice is there's not a whole lot of difference, and we don't see any of the points falling along the highest amount of cyclic we would expect to see. The other thing that you see is if we look really closely, I could show you that there's not a real significant difference between whether I gave them nitrate or whether I didn't give them nitrate. So not a real nitrogen effect. So what this tells us is that under low light, cyclic electron flux does not change with changes in energy demand. Now let's take a look at what I saw under high light. So here I've plotted it a little differently. So here, rather than looking at the slope of that line, I'm just showing you the slope. So this is proton flux per electron flux. When this value is high, we have a higher rate of cyclic. When it's low, we have a lower rate of cyclic. And I plotted it versus CO2 concentration. So I measured it under ambient oxygen and under low oxygen. So we can see, and then here's the modeled lines. So we can see that the modeled response of cyclic electron flux to decreasing CO2 concentrations is we see an increase as we move down in CO2. So as we move down in CO2, we have more photorespiration. Photorespiration requires more cyclic electron flux. We have higher rates of cyclic electron flux. The difference we see between these two is that under ambient oxygen, this response is fairly gradual. There's plenty of oxygen around to initiate photorespiration, so we see a gradual increase in cyclic electron flux with photorespiration. However, when we go to low oxygen, there's not a lot of oxygen around to initiate photorespiration, so we only have high rates of photorespiration until we get to very low CO2 concentrations. All right? So let's put in what I measured. So here we see that under ambient oxygen, we see what we would expect. We see gradually increasing rates of cyclic electron flux, about what we would expect if cyclic electron flux was operating to balance energy supply. When we go to low oxygen, we also see, again, this flat response that increases a lot right at the end. So both of these seem to support that under high light, cyclic electron flux balances energy supply and energy demand. The other thing we see is that there's not a significant difference between the different nitrogen forms we fed the plant. So from this, conclude that cyclic electron flux does shift with energy demand under high light. So the total outcomes from this last objective are that cyclic electron flux balances energy supply under high light. Under low light, other processes seem to be working to balance energy supply and energy demand. Finally, nitrate assimilation does not appear to play a large role in energy balancing. Maybe the energy is coming from a different source, or maybe rates of nitrate assimilation were just so low relative to photorespiration and CO2 fixation that they don't really factor in these type of calculations. All right, now we're going to summarize, and then we're going to summarize. So first we're going to summarize. Objective 1, photorespiration appears to lose efficiency with temperature. Objective 2, rubiscokinetics and mesophyll conductance respond differently to temperature between species. Objective 3, cyclic electron flux balances energy supply at high light, but not low light. All right, so great. I threw those objectives at you, but how does that come back to our original question? How will photosynthesis respond to increases in CO2 and increases in temperature? The answer isn't straightforward. All right? So while there'll be more CO2 available for photosynthesis, that higher temperature is causing more CO2 to be lost to the plant due to photorespiration. Additionally, there's species-specific kinetics that define how rubisco interacts with temperature to fix CO2 and to initiate photorespiration. On top of that, some plants are better at high temperature at bringing CO2 to rubisco to do photosynthesis. So we might see a competitive advantage of some plants that have a better mechanism to bring that extra CO2 in and do photosynthesis. On the plus side, we know that from this work that plants have a really robust way of balancing energy supply with energy demand. So whatever the CO2 is, they'll find a way to figure that balance out. So thank you all for coming. I'd like to thank my graduate committee, both past and present, past, as well as wonderful undergrads, especially Lauren, who helped do a lot of this work, the entire Cousins Lab, and especially Emily and our child Bridger, who has been providing the musical accompaniment for the evening. So with that, I thank you all for coming and would love to take any questions.
All right, questions for Mr. Walker?
Yes, Donna. Oh. Yeah. I mean, I first off, I have no idea at all, but I could make some guesses.
Is there a wild type tobacco?
Yeah, I don't know how different the wild type in the wild looks, because I mean, the Native Americans were cultivating tobacco for I don't know how many hundreds of years before we came. And I'd imagine the selection for big leaves does things, in terms of water use efficiency and nitrogen use. But I just don't have a good answer for that question. Yeah.
Like a Great Dane of tobacco.
Yeah. I will say that when you see field tobacco, it's a lot bigger than the tobacco we use in the lab. And so we call our tobacco wild type. I don't know if it's a true wild type, but it might be more similar to what's in the field, or what's in nature as opposed to what's grown in fields.
One of the major differences from the Green Revolution is really the partitioning of that carbon, where it's going. So a lot of selections then for plants that are smaller in stature, so there's more phenotypes, so the partitioning more resources to bring you in your flower reproduction, things like that. So not allocating it to biomass production unless it's for tobacco, for example, if that's what you're harvesting. So it's been a real shift in that direction as far as the selection of plants that have a more efficient capacity to put carbon where we want it to.
Thank you. Any other questions? Yeah.
So after doing all this research, do you feel more or less confident in the kind of global projections?
Yeah, I do a lot of thinking about it, and it's funny because you would think after doing this I would have a good tool set to go out and see, like, how can we apply this to the world? And it's just such a toss-up, because I've just looked at 2 species. Who's to say I can generalize to all warm adapted or all temperate? I mean, I think the best way to answer that is what I said at the very end, is that it's a complex picture. We're going to be losing more CO2 to photorespiration. There are going to be increases due to more CO2 for carbon fixation. How are they going to break even? I don't know. That's maybe something that Waldo would be better able to answer. She's more of a climatologist. Oh, yeah.
Mentioning the CO2 models, are those used more for yield projections, or is that used for climate models?
Yeah, so this is stepping a little bit outside of where I'm super familiar with the literature, so this is just kind of what I remember from what I've looked at. The model that I showed you is used more for global carbon cycle modeling. Because you have that during the day, but then at night you have this huge respiratory release of CO2. And so I'm sure people have scaled this up to yield models. I don't know how good they are for yield models, but I know they are used more extensively to scale up to see what canopies are doing in the under illumination in the day. I think there's another question kind of over here. Yeah.
I wonder if the peroxisome picks up other function as temperature increases that's not photosynthetic.
Yeah, I've thought about that because they have catalase mutants. So they have mutants where they have them overproduce catalase in the peroxisome. So with more catalase, you would take away that hydrogen peroxide and you may decrease this photorespiratory release, right? And that's something I'd like to look at. I think would be interesting to look at. The interesting things is that hydrogen peroxide is thought to be a signaling molecule in the plant, a stress signaling molecule. And so maybe the plant needs that extra hydrogen peroxide to really let everything know that, all right, it's getting warm, we need to start changing things in this way. And so it would be interesting to see if you lose out in the long run by minimizing that hydrogen peroxide. But beyond that, I don't know. That's just a speculation on my part. Any other questions?
All right, well, let's thank Berkley again.
Yeah, I worry where am I going? Some don't believe